Good morning to all of you. I am Ritu Gehlot from St. Andrews Institute of Technology and Management, Gurgaon. Today, I am taking my 15th lecture in which we are going to start our unit 3rd, that is the genetic engineering. So let's start our topic, or we say that is a new unit of our syllabus, that is the introduction of genetic engineering. So, as we know that genetic engineering is a biotechnology. In early days, we know that in early times this is called the biotechnology, and now it is called the genetic engineering. So, as we know that the biotechnology means it has the two words in there. The first word is the biology, and the second word is the technology. So it means and the meaning of biology. Everyone know that it is a living thing, and the technology, so which is related to the biology. It is biotechnology, or we can say that it is the control use of the living organism with the help of technique is the called biotechnology. So now come to the topic, and we know that biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce product and process useful to human. And as we know that in the early time, we know that the making the curd, bread, or wine, so which are all micro-medicated processes, and they could be thought as a form of biotechnology, but however, it is used to restricted sense today. So now, the word which is used as a biotechnology in the early times, and nowadays it is called the genetic engineering. Or here we have the so many definitions of biotechnology. So according to the new definition, the biotechnology means that the use of biological processes, organism or system of manufacture of products which are used to improve the human life. That is, yeah, improve the quality of human life is the biotechnology. Okay, so that means the हमें कहना क्या है कि ह्यूमन लाइफ को इम्प्रूव करने के लिए जो टेक्निक्स हम यूज करते हैं दीज आर द बायोटेक्नोलॉजी or genetic engineering. So here come to the term biotechnology again. So biotechnology can be defined as an application of technology using the living organisms to obtain useful product. It may be plant, it may be animal also. Okay, or we can say that biotechnology is the control use of biological agents such as microbes or the cellular components. So these are the definitions of the biotechnology. The modern biotechnology, which is the genetic engineering, here we can say that genetic engineering or modern biotechnology means this is the manipulates the genetic information as an organism. Yeah, gene modification is also called, or gene manipulation is called. So this is the modern biotechnology, or it is called the genetic engineering. Now come to the definition, proper definition of the genetic engineering. That it is the process by which piece of DNA are transferred from one organism to another organism. एक organism से जीन को डीएनए को निकाल के और दूसरे ऑर्गेनिज्म में ट्रांसफर करना, तो दिस टेक्नोलॉजी इज कॉल्ड द जेनेटिक इंजीनियरिंग। और वो का क्या हो सकता है कि इट में बी ह्यूमन टू बैक्टीरियम, बैक्टीरियम टू प्लांट भी हो सकता है, एंड ह्यूमन टू शीप भी हो सकता है। सो ऑल दीज आर द biotechnology or genetic engineering part. And here one example of biotechnology, that is the in vitro fertilization leading to a test tube baby. हमने सबने सुना है इसके बारे में. तो it is synthesizing a gene and using it, so that developing a DNA vaccine or the correcting of the defective gene, all are part of the biotechnology. So this is the biotechnology. Now come to the principles of biotechnology. So the two main principles are there, and both are the very very important. Here the two techniques that enabled birth of modern biotechnology are, so these are the first is called the genetic engineering. So in the genetic engineering, we know that the techniques to alter the chemistry of genetic material हम लोगों ने पहले पढ़ा है कि these are the DNA and the RNA, to introduce these into host organisms and thus change the phenotype of the host organism. एक से निकाल के दूसरे में डालना और उसको बिल्कुल चेंज कर देना, कोई भी डेफिशिएंसी है तो उसको इम्प्रूव करना, ये सब चीजें जेनेटिक इंजीनियरिंग का, these are the parts of the genetic engineering. The first part is the techniques to alter the chemistry of genetic material like DNA and RNA to introduce these into host organisms and thus change the phenotype of the host organism. And the second is by firstly we take the DNA from the one organism, and now the second part is the maintenance of sterile ambience in the chemical engineering process to enable growth of only the desired microbe in large quantity for the manufacture of biotechnology. Okay, biotechnological product like antibodies, vaccine, enzyme etc. कि पहले ले लिया फिर उसको स्टाइल करना है, ठीक है, क्योंकि ताकि उसके ऊपर जो भी अनडिजायर्ड माइक्रोब्स हैं उनका इफेक्ट ना हो, और जो भी डिजायर्ड माइक्रोब्स हैं उनकी दे आर प्रेजेंट इन द लार्ज क्वांटिटी फॉर द मैन्युफैक्चरिंग ऑफ बायोटेक्नोलॉजिकल प्रोडक्ट्स लाइक द एंटीबायोटिक्स हैं, वैक्सीन्स हैं, जो एंजाइम्स हैं, जो भी एटसेट्रा में बनाने हैं. So there are the two main principles. I repeat the principle again. First, because they are the very important. First is the genetic engineering. In this, the techniques to alter the chemistry of genetic material. These are the DNA and the RNA, to introduce these into host organism and thus change the phenotype of the host organisms. So this is the genetic engineering. And the second part is that to maintenance of sterile ambience in chemical engineering process to enable growth of only the desired microbe in large quantities for the manufacturing of biotechnological products such as the antibiotics, such as the vaccine, enzymes, etc. And now uh, here is the definition of the biotechnology or in genetic engineering according to the European Federation of Biotechnology. So according to the European Federation of Biotechnology, इसमें से EFB, that the integration of natural science and organisms, cells, parts of thereof, means there are the compounds, these compounds are, and molecular analogus for the product and services. Means ऐसा नहीं है कि हमें एक ही branch चाहिए, तो there are the all branch which is related to the science और natural science, और ऑर्गेनिज्म हो, सेल्स हो, कोई भी उसका पार्ट कंपोनेंट हो, तो उनमें हमें चेंजेस करके उसकी प्रोडक्टिविटी को इंक्रीज करना. So that is the definition by the European Federation of Biotechnology. It is important. The integration of natural science and organisms, cells, parts thereof, and molecular analogus for product and services. So it is also important. And now here are the some definitions or some terms. We can say that these are the terms which is used in this like in this topic uh, time to time. So first is the word is gene. What is a gene? So gene is the small piece of DNA that encodes for the specific protein. Okay, है ना. तो जीन क्या है? वो small piece of DNA है that encodes for the specific protein. Second is the very very important term that is called the recombinant DNA. So here uh, recombinant DNA is also denoted by the rDNA. So the DNA formed by the joining DNA segment of two different organisms. यहाँ पे एक चीज क्लियर करनी है कि एक से एक किसी हेल्दी प्लांट हो चाहे एनिमल हो चाहे किसी से भी हमने डीएनए को लेना और फिर दूसरे के साथ उसको ज्वाइंट करना. तो और आफ्टर दैट जो डीएनए तैयार होके आएगा हमारे पास, दैट इज कॉल्ड द रिकॉम्बिनेंट डीएनए. So it means the DNA formed by the joining DNA segment of two different organisms. Okay, so this is called the rDNA or recombinant DNA. Now the third is the recombinant DNA technology. So in the इसमें the technique by which gene of interest is transferred to the host. जो तैयार होके आया है रिकॉम्बिनेंट डीएनए, अब उसको ट्रांसफर करेंगे होस्ट में. सो द टेक्निक बाय विच जीन ऑफ इंटरेस्टेड is transferred to the host. Now next term is that is genetically modified organism. It is called GMO. So the organism whose genetic makeup is altered or changed using recombinant DNA technology. Clear? So GMO, the organism whose genetic makeup is the change using the recombinant DNA technology. So these are the some important terms which are used. So these are the important terms which are used in our chapter time to time. So now, what is the method in the uh, formation of the recombinant DNA, or we can say that is the genetic engineering. So here are the three main method. These are the firstly the formation of the recombinant DNA. The first method is the formation of recombinant DNA. So here how can we form the recombinant DNA? In our next lecture will be discussing. And the second topic, second method is the, or the second part of these are the gene transfer, and the third is the gene cloning. So first step में हम लोग क्या करेंगे? Here is the formation of recombinant DNA. Firstly, there is the formation of recombinant DNA, recombinant DNA. Then recombinant DNA is here is a gene transfer, and the third is the gene cloning. So this is the method, and now come to the procedure. So here the procedure which is covered in these steps. So firstly, what we do, first point is that the isolation of desired DNA. जो हमें डीएनए चाहिए, तो आइसोलेशन ऑफ डिजायर्ड डीएनए फ्रेगमेंट विद द हेल्प ऑफ restriction enzyme. Okay, so the in the procedure, the first point is the isolation of desired DNA fragment with the help of restriction enzyme. Second point is the isolation of DNA vector. Then the third point is the constriction of recombinant DNA, and in this gene of interest is inserted into the vector. This is the third point. Now come to the fourth point, that is the introduction of vector containing recombinant into the host cell. These are the factors, or we can say these are the method, or we can say these are the steps. And then the multiplication of the host cell containing rDNA, and then the expression of colon's gene, and then the selection of the recombinant cell. So here these are the process. This is the process. I repeat again the process. First is the isolation of desired DNA fragment with the help of restriction enzyme. And then the second is the isolation of DNA vector. Third is the constriction of recombinant DNA. In this gene is interested is inserted into the vector. Then introduction of vector containing recombinant into the host cell. Then the multiplication of host cell containing recombinant DNA, then the expression of colon gene, then selection of the recombinant gene. So these are the this is the process step by step which we will discuss in our next lecture. So because today is the introduction of the genetic engineering, and then here are the some information. So I want to discuss with you that first step, as I told, the first step is the recombinant DNA formation. So here we want to say that I want to say that कि the recombinant DNA का formation किस तरीके से होता है. Okay, so the first step is the here we can see that there is a plasmid. ओके, अब प्लाज्मिड क्या होता है भाई. यहाँ पे कुछ टर्म्स हैं जैसे प्लाज्मिड है, फॉरेन जीन है. सो हम पहले हम अगर प्लाज्मिड की बात करें तो इन द प्लाज्मिड क्या है कि इट इज अ जेनेटिक स्ट्रक्चर इन अ सेल that can replicate independently of the chromosomes. Clear? That means there is a genetic structure in the cell that can replicate independently of the chromosomes. So typically, a small circular DNA strand in the cytoplasm of a bacterium or the protozoan, but the plasmid are much used in the laboratory manipulation of the gene. So this is the plasmid. But here we can say that the structure of the plasmid, and this is the another genetic material. ठीक है. अब क्या होगा कि there is the joining of the two plasmid and the genetic material. So first here is the plasmid, uh, cut of the plasmid, and then this the foreign gene which comes from the another genetic material, so that these are the joined together and they are the formation of the recombinant DNA. How can be the recombinant DNA formed? So this is the very simple, according to the definition, very simple or point, that the first is the plasmid and the other genetic material. And firstly, but this one is a part of the only one this part. Suppose हमें यही part चाहिए तो हम इसी को लेंगे, है ना. So this part is the foreign gene, comes from the genetic material, and it is called the foreign gene. Here is the plasmid, and then when they are combined together, then is the formation of the recombinant DNA. And the according to the our method, that is the second point, that is the gene transfer. And here is the gene transfer is, this is the recombinant DNA as we see in this picture. The first is the plasmid, this part is the comes from plasmid, and this is the foreign gene. So here we can see that is the second part is the gene transfer. So firstly this this here we can see that part is comes from the this is the plasmid part and this is the foreign gene. So they combined and they form the recombinant DNA. And here is the another genetic material of the host. So it is the genetic DNA from the host, and it is the plasmid. Okay, then ये किसमें है, ये आपका host bacteria. ठीक है. अब इसके बाद थर्ड स्टेप क्या है हमारा, दिस इज द जीन क्लोनिंग. अब क्या होगा कि जो प्लाज्मिड तो जो ये रिकॉम्बिनेंट डीएनए है अंदर आ गया होस्ट सेल के, अब उसको मैनिपुलेट करना है, मींस नंबर इंक्रीज करने हैं. सो इन द जीन क्लोनिंग देयर द फॉर्मेशन ऑफ द अदर recombinant DNA or other gene. So this is the gene multiplication. So So here there are the method. I repeat again the method. First method is the formation of the recombinant DNA, in which there is a recombinant DNA is formed by the plasmid and the other foreign gene. So when they are combined together, then they are the formation of the recombinant DNA. In this diagram, in this picture, we shows that the blue part is called represent the plasmid, and the second red part is the foreign gene. And then the second part is the gene transfer. So the transfer of the gene in the bacteria. And then the third step is the gene cloning. So in the gene cloning, there is the multiplication of the gene. It's called gene cloning. Now come to the tools, which is very important. Okay, tools. Write the tools which are for the recombinant DNA, important for the recombinant DNA. So here the tools, and this is the tools of recombinant DNA technology. It is a very important part of this lecture. So here, uh, one for first point, that is called restriction enzyme. Okay, restriction enzyme, or it is also called the cleavage enzyme. क्लीवेज ऑल्सो कॉल्ड, जिसका मतलब क्या हुआ कि इसका काम है तोड़ना, है ना, रिस्ट्रिक्शन एंजाइम और क्लीवेज एंजाइम। और ये कहाँ से तोड़ेगा वो अब हम इसमें डिस्कस करेंगे। तो फर्स्टली द रिस्ट्रिक्शन एंजाइम क्या कह रहा है कि रिस्ट्रिक्शन एंजाइम बिलोंग टू अ क्लास ऑफ एंजाइम्स कॉल्ड एज न्यूक्लियस। So these enzymes can cut the DNA double helix at the specific sequence. क्लियर, कि एक स्पेसिफिक जगह से जहाँ पे हमारे सारे न्यूक्लियोटाइड लगे हुए हैं, जैसा कि हम पहले डिस्कस कर चुके हैं लेक्चर्स में, तो वहाँ उनमें से टेक अ स्पेसिफिक सीक्वेंस, है ना। तो द रिस्ट्रिक्शन एंजाइम cut the DNA. DNA कैसा है? That's a double helix structure. So cut the DNA at the specific sequence, or which is depend upon the there are the two types of the restriction enzyme. Firstly is called the exonuclease, and second type is called the endonuclease. So here is the definition with the structure. So here we can say that the exon. Firstly, we will talk about the exonuclease enzyme. So in the exonuclease enzyme, these enzymes removed nucleotide from the end of the DNA. I repeat again, ये कहाँ से कट करेगा कि the enzyme removed या cut nucleotide from the end of the DNA. एंड साइड से डीएनए को सपोज से हम देख लें यहाँ पे कि हमारे पास दिस इज द डीएनए स्ट्रक्चर ऑफ डीएनए, एंड एज वी नो देयर डीएनए इज द डबल हेलिक्स स्ट्रक्चर, तो इट मींस कि देयर आर द टू strands are present. First is here we can say that is the 5 prime to 3 prime, and this is the 3 prime to 5 prime. This is here the nucleotide are present. So this is the end of exonuclease, and now here we can say that the enzymes cut the nucleotide from the ends of the DNA. So here we in the second part we can say that the cut कहाँ से किया, this part एंड साइड से, चाहे वो कहीं से यहाँ से भी कर सकता है यहाँ से भी कर सकता है, तो एंड पार्ट से इसको कट किया। तो अब जो ये एंड पार्ट है दिस इज कन्वर्टिंग टू द 3 prime to 3 prime. Okay, and when we talk about the endonuclease, so in these make the cut at specific position within the DNA. इसके exonuclease enzyme कहाँ से कट कर रहा है? At the ends of the DNA. But endonuclease enzyme कहाँ से कट करेगा? At the specific position within the DNA. कहीं बीच में से कहीं से भी, लेकिन these make the cut at the specific position within the DNA. These enzymes recognize specific sequence. These enzymes recognize specific sequence are called palindromic nucleotide. Okay, it is called the palindromic nucleotide sequence in the DNA. अब palindromic nucleotide क्या है ना? यहाँ पे हम बात करें पैलिंड्रोमिक न्यूक्लियोटाइड का मतलब है कि जो दोनों साइड से सेम होगा, ठीक है, कि जो न्यूक्लियोटाइड है उनको कट करा जाए तो वो दोनों, सपोज हम एक स्पेलिंग लेते हैं, टेक द नेम नितिन, तो नितिन इज द सेम, दोनों साइड से आप कहीं से पढ़ लीजिए वो नितिन ही रहेगा, है ना। ऐसे ही और अनदर वर्ड वी कैन टेक दैट इज द मैडम, so the spelling of the madam is the another example of the palindromic nucleotide, palindromic. तो इन द पैनिड्रमिक मीन्स कि वो दोनों साइड से सेम ही होगा, है ना। एज अनदर वर्ड इज मलयालम, दिस इज द अनदर वर्ड। सो मीन्स आगे से पढ़ो या पीछे से पढ़ो, वो दोनों की स्पेल दोनों मतलब सेम होना चाहिए, है ना। सो दीज मेक द कट एट स्पेसिफिक पोजिशन विद इन द DNA. सो दीज एंजाइम्स रिकॉग्नाइज्ड स्पेसिफिक सीक्वेंस एंड कॉल्ड द palindromic nucleotide sequence in the DNA. This is the endonuclease enzyme. Here we can see this is the DNA structure, और ये कहाँ से कट करेगा? बीच से कहीं से भी कट कर सकता है, जहाँ से भी इसको आपका palindromic nucleotide sequence मिल जाएगा, ठीक है। तो this is the endonuclease. Now there are some examples of the restriction endonuclease enzymes. So here the first is a ECOI1. It is obtained from the E. coli. E. coli is a bacteria that is called the name Escherichia coli, and it is recognized here the sequence GAATTC है। तो उसका जो कॉम्प्लीमेंट्री जीन्स बना वो हमारा हो गया अपोजिट। जी में हियर इज द ग्वानिन, ग्वानिन के साथ साइटोसिन, एडेनिन goes to thymine, adenine, thymine, thymine, adenine, thymine, adenine, cytosine, guanine. So here the cut one strand is 5 prime to 3 prime, another is a 3 prime to 5 prime strand. Here we can see the special sequence. It means that this enzyme cut the nucleotide from the between the G and A nucleotide. So where the G and A are present, so it will have the cut of this at this sequence. Okay, तो यहाँ पे G और A है तो ये यहाँ से इसको कट करेगा, ठीक। And the second type of the example is BAMHI. So this is obtained from the Bacillus amyloides नाम fancy जो भी है, and these are the recognized as the GGATCC. And here's the again GC, it कहाँ से कट कर रहा है between the GG. Okay, this is the sequence. This is the GG. Okay, so it is these are the examples of the palindromic. हाँ, this is called the palindromic nucleotide. तो दोनों साइड से वो सेम है. Now the third is the hint 2. It is the firstly first it is the first restriction endonuclease hint 2. So it is here the hint 2 is obtained from the H influenza, and it is a sequence. It is the AAGCTT. Okay, so here again the TTGCGAA, because A goes to T, A with T, G with C, C with A, G, T with A, T with A. So there the cut between the A and A. Okay, जहाँ भी आपके दोनों A है nucleotide, तो here you can cut. Okay, then the next type of the endonuclease enzyme is the TAKQL, that is obtained from the thermus aquatics क्या है aquatics, and it is a TCGA, and again GAGCT. Okay, so these are the palindromic nucleotide, and the cut between the T and C, जहाँ भी T और C होंगे तो वहीं से ये cut होगा. So these are the type examples of the restriction endonuclease enzyme. Okay. So it is our first lecture of the genetic engineering, in which we have uh, discussing the general information or introduction of the genetic engineering, which is another word of biotechnology. And biotechnology is the word term which is used in the earlier time, and now it is called the genetic engineering. And here are some other different definitions and the principles of the biotechnology. Then we goes to the according to the European Federation of the Biotechnology. Then some terms like gene, recombinant DNA, recombinant DNA technology, genetically modified and organism, and it is the methods formation of the recombinant DNA, then the gene transfer, and then the gene cloning. And now next to the procedure, there are the steps, and then here is the diagrammatically explanation of the recombinant DNA formation, then the gene transfer and the gene cloning, and here are the tools of recombinant DNA. Today we have discussed only the one tool, that is the restriction enzyme, and there are the so other many tools are also which we'll be discussing in our next lecture. So in the restriction enzyme, there are the two types of the restriction enzyme. First is the exonuclease, and another type is called endonuclease. And here are the some examples of the restriction endonuclease enzyme. So now I stop complete my lecture, and you can study the notes, and if you have any query, then contact with me. Thank you.